Assalamualaikum and good day everyone. I am Suzana Misbah. Today, I am going to talk about mosquito immune responses. In the previous video, you have learned about the biology of Aedes mosquitoes and several ways of controlling these vectors. Now, in this video, we are going to explore another interesting topic in dengue and Zika vectors, the mosquito immune responses. Despite decades of vector control efforts, dengue and Zika remain a significant public health threat in large regions of the world. Dengue causes an estimated 400 million infections annually, with 3.6 billion people living in areas at risk for epidemic transmission. Meanwhile, Zika has not been widely reported, which could be due to asymptomatic cases, clinical resemblance to other virus infection, and difficulty in confirming diagnosis. The main mosquito vectors transmitting dengue and Zika viruses are Aedes aegypti and Aedes albopictus. As natural vectors, these mosquitoes are very permissive to persistent virus infection, which allows them to transmit the viruses efficiently. Such infection in the mosquito tissues, however, does not result in any pathological significance in the mosquito or affect the mosquito lifespan. How does this happen? This is due to the fact that mosquitoes have efficient antiviral strategies which are able to tolerate persistent infection and restrict viral replication. Interesting, isn't it? So these strategies lies within the mosquito innate immune responses. So in this topic, we are going to learn about mosquito innate immunity, by which mosquitoes control viral infection in their tissues. After you have finished watching this video, we expect you to be able to understand 1. The process of how virus infects and disseminates in its mosquito host. 2. The establishment of mosquito cell lines as a tool for virus research. And 3. The interplay of mosquito antiviral defense mechanism during viral infection. So in trying to achieve those learning outcomes, we are going to cover a few main topics. They are dissemination of viruses in mosquito, establishment of mosquito cell lines, and mosquito innate immunity. Before we move on, let's have a quick look at the definition of arbovirus that you may hear in this video. Arbovirus, or arthropod-borne virus, refers to any viruses that is transmitted by arthropod vectors such as mosquitoes, ticks, or sandflies. The example of these viruses are dengue, Zika, yellow fever, West Nile, and tick-borne encephalitis viruses. So as we know, dengue and Zika are spread in a transmission cycle between mosquitoes and mammalian hosts. Humans contract dengue and Zika diseases through the bites of female Aedes mosquitoes on the skin. While seeking a blood meal, mosquitoes probe for blood vessels in the skin with their proboscis. once a blood vessel is pierced, the mosquitoes continuously deposit saliva while drawing out blood. The saliva contains many biologically active components, including anticoagulant and proteins. Have you seen a mosquito bite? It typically looks like a red bump and feels itchy. Most mosquito bites are harmless, but they are very dangerous for transmitting certain arboviruses, including dengue and Zika viruses. Before we move on to discuss about the mosquito immune responses, it is very important for us to know how virus infections spread in mosquitoes. During a blood meal by virus-infected Aedes mosquito, the virus is ingested and delivered to the midgut where the blood meal is digested. This is where the virus replicates and traverses into the lining epithelial cells of the mosquito midgut. Once it reaches sufficient titers, the virus escapes from the midgut into the hemolymph and subsequently spreads via the hemolymph circulation to other organs such as the fat bodies, nerve tissues, muscles, and finally the salivary glands. Here, the virus is secreted into mosquito saliva, which can then be injected into another host when the mosquito next takes a blood meal. In order for the virus to be successful for transmission to a new host, they must first overcome a number of restriction barriers in the mosquitoes. First is the midgut infection barrier, which impedes virus access to the mesenteral cells and prevents replication. Second, the midgut escape barrier that restricts the movement of virus particles from the midgut to the surrounding tissues in the hemocoel. Virus, thus, must be in very high titers and pass the required threshold in order to overcome these physical barriers. Virus progeny that makes it to the salivary glands are then subjected to the salivary gland infection barrier and the salivary gland escape barrier. If this is successful, the virus will replicate in the salivary glands to higher titers than in any other tissues for exportation by the saliva. Transmission of dengue and Zika diseases requires the viruses to undergo a significant period of time within the mosquito vector, where the viruses replicate and finally disseminate to the salivary gland. This is called the extrinsic incubation period, which is the time taken between ingestion of a blood meal and the ability of mosquito to transmit viruses. For example, dengue virus extrinsic incubation period is typically between 8 to 12 days, and this is also influenced by external factors such as temperature. The intrinsic incubation period is the time between a human being infected by the viruses and the onset of the symptoms due to the infection. This takes approximately 3 to 14 days. Human can become ill after this incubation period. Now, how are we supposed to study diseases caused by arboviruses? So this requires the development of mosquito cell cultures. There are over 100 dipteran cell lines now derived from different insect tissue sources. Aedes mosquitoes are among the most common insect cells used as sources of cell cultures. The mosquito cell lines have been established for virus propagation and production of recombinant proteins. These include various molecular studies such as virus-host interactions, pesticide resistance, immune responses at the mosquito cellular level. Early establishment of the mosquito cell lines begins since the mid-1960s. CCL125, a mosquito cell line derived from larvae of Aedes aegypti, has been developed, which is permissive to dengue virus infection. Subsequent cell lines derived from Aedes aegypti, including the AAG2 and RML12, have been later developed for the study of mosquito immune system. The first cell line developed from Aedes albopictus is C636 cell line, which are generated from the mosquito larvae. This cell line is susceptible to a wide range of arboviruses, particularly due to the lack of a functional RNAi immune response, and has been widely used to isolate arthropod-borne viruses. The C710 cell line was derived from Aedes albopictus and has been shown as a useful model for understanding hormone responses in insects. Now let us move to look at the general mosquito immune system. As mosquitoes transmit a number of medically important viral pathogens, such as dengue and Zika, understanding their antiviral immune responses has been essential to disease control. Mosquito immune system is different from those of mammals. Unlike mammals, which have both innate and adaptive immune systems, mosquitoes lack the adaptive immune responses and instead rely heavily on their innate immune defenses to modulate viral infections. These mechanisms are able to tolerate the viral persistent infection and restrict their propagation to non-pathogenic levels. In other words, virus infections in the mosquito vector is successfully controlled by the antiviral response, thus will have no detrimental impact on the mosquito's survival. As we have known, transmission of dengue and Zika viruses occurs through the bite of a mosquito vector to a mammalian host. Hence, the viruses now have to overcome two very different host immune systems, which are the mammalian immune system and the immune system of the mosquito vector. Until now, very little information is obtained about the immune responses that control virus replication in mosquitoes. Scientific evidence indicates that there is a complex interaction between the virus and the mosquito immune system, which actually controls the virus infection and dissemination in the mosquitoes. The first step of any signaling pathway in the mosquito is the recognition of the pathogen through pattern recognition receptors, which actually identify foreign entities by its pathogen-associated molecular patterns, or PAMs. PAMs are conserved components of different infectious agents, such as the viral nucleic acid, bacterial cell wall components, or fungal complex carbohydrates in the cell wall. Following pathogen recognition, the host signaling pathway relays the information to stimulate an effector response through the transcriptional activation of certain genes. In mammals, these genes mostly constitute those which are interferon dependent and encode for molecules such as interferons, chemokines, cytokines, and antimicrobial peptides. However, in mosquitoes, antimicrobial peptides such as defensin, attacin, and drosomycin are produced in the mosquito fat body, which functions as the equivalent to the mammalian liver, and these are secreted into the hemolymph in very high concentrations to facilitate immunomodulation against the invading pathogen. There are three main signaling pathways that may be activated in response to virus infection in mosquitoes. They are TOL, immune deficiency or IMD, and JAK-STAT pathways. Other cellular pathways play a role in modulation of virus infection, including melanization, apoptosis, and autophagy. I am showing you here the overview of innate immune pathways in mosquitoes. Much of what is known about these pathways has been established in Drosophila melanogaster as the model insect organism. Activation of these pathways may also result in the expression of antimicrobial peptides, which can be antiviral. How viruses activate these pathways are still poorly understood. However, they have been shown to be able to modulate dengue and Zika virus infections. Toll pathway stimulates activation of antimicrobial peptides and target fungi and gram-positive bacteria. In this pathway, detection of PAMs initiates proteolytic signaling cascade, resulting in the cleavage of Spetzel, a dimeric cytokine. Active Spetzel binds to the toll receptor, which induces dimerization and triggers the recruitment of downstream intracellular proteins. This leads to the phosphorylation of the negative regulator cactus signaling for its degradation. Following this, a protein called RELISH1 is able to translocate to the nucleus and activates the transcription of effector genes. IMD pathway is activated by gram-negative bacteria. Detection of peptidoglycan by peptidoglycan recognition proteins-like receptors triggers this pathway and leads to the cleavage of another protein, RELISH2, which translocates to the nucleus and initiates immune gene transcription. JAK-STAT pathway is also activated by gram-negative bacteria. This pathway involves the cytokine VAGO that binds to an unknown receptor, activating the Janus kinase, or JAK, which in turn phosphorylates signal transducers and activators of transcription, STAT. STAT is then dimerizes, translocates to the nucleus, and transcribes the target genes. In addition to the three immune signaling pathways, mosquito has an effective antiviral mechanism called the RNA interference pathway, which is a sequence-specific RNA silencing mechanism thought to be the main immune pathway that plays a key role in antiviral defense. Although there are many pathways within RNAi, such as the microRNA and PIWI interacting RNAi pathways, I shall explain about the exogenous small interfering RNA, as this has shown to be the major component of the mosquito antiviral response system. The exogenous siRNA pathway is initiated by an enzyme called Dicer2 through the detection of long double-stranded RNA molecules resulted from RNA virus replication. Dicer2 has an endonuclease activity that cleaves the RNA molecules into small double-stranded RNA fragments of 21 nucleotides in length called exo-siRNAs or virus-derived siRNAs. These fragments are recruited into a ribonucleoprotein complex called the RNA-induced silencing complex, or RIS, containing several components, including the organoid protein AGO2. Further components involved in the assembly of the active RIS include component 3 promoter of RIS, or C3PO, aubergine, and heat shock protein. Once incorporated into the active RIS, the siRNA duplex is unwound and one strand is degraded while the other strand is used by AGO2 as a template to identify complementary mRNA molecules or virus genomes, which can then be degraded. The slicing of AGO2 results in the sequence-specific degradation of RNA molecules and thus silencing of target proteins. For the virus, this means the inhibition of viral gene expression and a reduction in the viral progeny produced. As a summary, we had discussed about how the virus infects and disseminates in mosquito tissues, the types of mosquito cell lines established for arbovirus in vitro studies, and the mosquito antiviral defense mechanisms against virus infections. The understanding of mosquito antiviral mechanisms, however, is still unclear. Major challenges including identifying of molecular events of the antiviral immune system, the virus sensing machineries, and their mechanisms of action remain poorly understood. Further investigation of the crosstalk between the known antiviral pathways are needed, which will provide insights into the interactions between mosquitoes and arboviruses. To conclude this topic, we learned that the mosquito antiviral defense system may not fully eradicate arboviruses, but they can actually effectively limit viral propagation to a tolerable level without detrimental consequences to the mosquito. This involves complex interactions between the mosquito immune system and viruses, which makes mosquitoes an ideal transmission vector in nature. We also have learned that RNAi, Toll, IMD, and JAK-STAT immune signaling pathways are very important in controlling arboviral replication and dissemination in mosquito tissues. As this topic focuses on defense mechanisms in mosquito hosts, in the next topic, you will be able to learn about the mechanism of how our body, as a human, controls virus infection and all mechanisms involved to fight the virus infection. So, till next time, see you around, guys. Thank you.